Hello, friends. Good morning to all of you. So, today I would like to discuss and share very interesting biological question. That biological question is whether mitochondrial defects influence or affect the severity and fatality by SARS-CoV-2. In a very simple way, consider if any patients are exposed to SARS-CoV-2 and that patients they have either inherited mitochondrial defects or the patients have the secondary mitochondrial dysfunction like in case of cancer, diabetes and many more. So whether these patients populations where the either primary mitochondrial defect in the form of genetic or inherited disorders or secondary mitochondrial dysfunction may influence the severity and fatality by SARS-CoV-2 that we know that is the COVID-19 as a you can say broad term given to this type of the infection. Then potentially answer should be yes. So I believe that this type of the answer or the understanding will add or you can say will complement to the thousands of research papers those are published in the recent times. Okay, friend. So the whole interesting point is if the mitochondrial defects are there, and most importantly, I would like to discuss here the lung epithelial cells, that is the host cell or the target cell for SARS-CoV-2, where the spike protein, that is receptor binding domain of spike protein that binds to the ACE2 receptor on the host target cell. Then what? How the mitochondrial defect within these lung epithelial cells, that is the host cell, may be connected with the severity and fatality by SARS-CoV-2 infection. That is the whole question here. That is fine. So in this direction, I would like to discuss two model and the proposed model where it is scientifically connected, and that's what I would like to share these particular views here. Okay, dear friend, if you look at here, this is the model of where we can see if the mitochondria is defective, whether in the form of primary mitochondrial defects, inherited, genetic, or secondary mitochondrial dysfunction. So, what is going to happen? If you look at the model here, this is the the SARS-CoV-2, which is going to adhere to the membrane of lung epithelial cells, and this is our spike-2 protein through the RBD. They are going to attach to this particular membrane. So, this is the membrane, okay, and here is the ACE2 receptor sitting on the membrane. That is well known. There is no any doubt. So what is happening here? Once the whole bound complex, they are going to be go through the endocytosis and they are going to enter to the cytoplasmic world of these host cells. So then what is the scene here? What is happening here? So the cytoplasmic world, or you can say the cytoplasmic compartment of host cell, whether they are doing something, what is the environment inside the cytoplasmic compartment of these host cell where the SARS-CoV-2 has, or you can say the SARS-CoV-2 are able to bind and they are able to enter to the cytoplasm. So our view is here very clear. If there is a mitochondrial defect, if you look at here, this is the mitochondria, this is the mitochondria, and there may be few to several hundreds of mitochondria present in this lung epithelial cell. So the question is always there, if the mitochondrial defects will be, that means the ATP production by these mitochondria will be also compromised. As per the literature, what I know, that if the mitochondria is very highly active, cells are healthy, no defects are there, so the if you look at the surrounding of the mitochondria, the temperature is maintained at 50 degrees Celsius. Surprisingly, and these tiny mitochondria, they are very heated or high temperature, so the heat are going to be dissipated to the other part of the cytoplasmic compartment. And here look at, if the mitochondria is defective, that means the 50 degree Celsius and the heat dissipation to the cytoplasmic compartment will be compromised. Then what? So what we believe, if the temperature within the intracellular compartment of the host cell where the SARS-CoV-2 has entered, then these SARS-CoV-2, they will find conducive, friendly intracellular compartment environment where these SARS-CoV-2, they can replicate, then translation will be there, uh, assembly of these virus particle at the end, and then many more virus particles are generated, and then ultimately you can see that is the whole consequences, that is in the form of severity and fatality by SARS-CoV-2 infection. Look at, so there are various factors that can influence severity and fatality. What we are proposing, that one of the factor can be the mitochondrial defects, where the temperature within the intracellular compartment becomes highly conducive and friendly for the SARS-CoV-2 multiplication inside the cytoplasmic compartment. That is the point here. And that's why we feel that severity will be more if any individual, they have their mitochondrial defects, either primary, inherited, or secondary infection. Great. Now I would like to show here another model where the mitochondria is healthy. Look at here. So if the mitochondria is healthy, then as I suggested, the close to the mitochondria here, look at this portion here, the temperature is very high, 50 degrees Celsius, or approximately. So these temperature, the other compartment of the cell, if you look at the cytosol, the fluid portion there, the heats are dissipated. So like this is the mode of heat dissipation. Look at here. So due to the dissipation of the heat, the intracellular compartment will be slightly and significantly higher in comparison to the where the mitochondria is defective. That is the whole interesting point here. That means if the mitochondria is, if the temperature within the compartment is higher side, we know that the various component of the SARS-CoV-2 protein, the virus particle, including spike 2 or many other protein like proteases, etc., they are highly temperature sensitive. So if the temperature of the intracellular compartment is maintained at a significantly higher side, that is definitely supported by the very functional mitochondria, then we believe that in that case there will be a little bit less severity, definitely in the form of less multiplication of SARS-CoV-2 virus particle inside the intracellular compartment of host target cell, that is the lung epithelial cell. That's great. So what is the conclusion here? I would like to relate with two clinical example. Look at here. So the clinical example one. So there are very recent research paper published in Science Journal. So where they have mentioned that if the SARS-CoV-2 exposed patients, they have the mitochondrial inherited disorders, so then comorbidity is very high. Severity of infection by SARS-CoV-2 is very high. The reasons may be various, but one of the reason can be the defective mitochondria is not able to defective mitochondria. It compromises the ability of the cell to defend, or you can say to do something. Another very clinical example is here secondary mitochondrial dysfunction. If you look at cancer as an example, so there is a very recent example or very interesting paper where the scientists have concluded that the comorbidities in case of cancer and SARS-CoV-2 is a little bit on higher side, high severity and fatality if the both cancer patients are going to be exposed by SARS-CoV-2 viruses. So, my dear friend, this particular observation and you can say the viewpoints may lead to certain understanding what should be done, what should be the preventive measures for a certain selected group of patients where the primary mitochondrial dysfunction is there, secondary mitochondrial dysfunction is there. So those patients or group of individual, they should be given proper attention, because these group of patients, they are more compromised to the SARS-CoV-2 infection, and that is in the form of COVID-19. Thank you very much. Drop by drop, ideas will create a good creation that is in the future. Thank you very much.